So we can actually annotate them based on the phenotypical characters, and so that they separate very well, or we can at least understand do they separate very well according to phenotypical characters or different sample characteristics that we associate from the for the data, etc. So before that, we can also enable loadings, which will tell you what are the important dimensions or what are the significant dimensions that contribute the most to separate the samples into these multiple groups. We observe that there are some groups that we can observe in these samples, and if you enable loading, we can see that there are some specific genes like this 160180 and 065618, and we can understand that these genes or these transcripts are responsible for isolating the samples to this group, and while these genes are responsible for isolating the samples to this group. And so hence we can gain more information, not about how or where the samples are grouped, but also what are the most informative genes in our dimensions or in our input table. So we can expand the principal component analysis not only to 2-dimension, but also to 3-dimension. In some of the cases, that will be very helpful, like how you interacted with the interactive 3D plot that I just demonstrated you. And in addition to that, as I mentioned, when you have annotation available, or when you have the sample characteristics available for your input table, we can load those sample characteristics also into a separate object, and we can use those sample characteristics to color the samples and color their distribution in the principal component analysis output, so that we can understand whether the PCA's variance explained actually separates the samples according to the different characteristics that we associate the samples with. We can extract these sample characteristics from biological condition under which the samples are measured, or different phenotypical characteristics of the sample as such. For example, if it's a gene expression data from a tumor sample, then we can take whether these samples are from what subtypes of tumor, or whether these samples are from tumor samples or adjacent normal samples, and any other layer of molecular characterization or histological characterization has been performed on these samples, and whether we can associate that to this sample. Or you can explore a little bit beyond those molecular characterization of the samples to the clinical characteristics of the sample, like the demographical distribution, like age group, like ethnicity, whether they are actually represented in the gene expression data distribution of our analysis, right? So how do we do that? So we actually now again go through some of the important codes that we already had some introduction to. So we load the packages. Some of the GG45 is one of the package that will let us use autoplot function that we have discussing so far. And in this read.table function, as I had told you, we specify the location of the sample here. And then we specify some of the characteristics that is associated with the sample, like the separator, like whether or not the first row mentions the header. And then we can add some associated options which will help us organize the data, like how characters to be interpreted, and whether or not do we have to check for duplicate row names, and etc. So if you load this data, and then if you check first few lines of the data, we can quickly find out that the first column is, okay, the first column will actually give you the annotation of the sample, and we can extract only the numerical component of the sample by selectively by selecting those specific numbers of column that we want to extract, and then we can apply principal component analysis only on the numerical component of this column. And if you do not do that, then this analysis is going to throw you an error that we have a non-numerical character or non-numerical counterpart input in your data. And here we enable scaling to be true, and immediately again we can use some basic functionality like autoplot to plot it. And in the autoplot function, as I was mentioning in the above section, we can specify, or we can actually include the annotation or sample characteristics that is associated with each and every sample. We can color the sample according to their annotation that we have included in the input table. So in this case, we are going to plot the principal component analysis result using autoplot function, and we are going to mention where this annotation is coming from. It is coming from the data table data, and the column that gives you the annotation is group. And when we do that, we can immediately see that the samples are colored, and our analysis of this input data reveals that we have very good separation of different sample types, like the normal, like luminal, and all the 4 subtypes of this tumor. They are very well separated, as you can see, and that is very good, very nice to know. And we also find out that the principal component analysis actually covers quite a bit of our very good range, or explains a very good percentage of variance in the first 2 principal component themselves. Like the principal component 1 explains 50% of the variance that you find in the original data, and together they explain something around 68% of them. And in addition to that, again, we see that we can actually label them, and there is, okay, we can actually label them according to the sample type, and we can also we can label them according to the sample types, and this label has to be sample type. There is some error, then I will take care of that. So once you learn about how you can perform different principal component analysis, and how various different visualization options will help you understand the output of this analysis, and you can fine-tune the output or fine-tune the visualization for your need, for your necessary. Maybe you need only up to this point, or maybe you need to include the sample types or sample labels along with this output. So you can do that with the given options that is available. So once you have learned this code, so the next area or the next section in this lesson will help you practice all the codes that you have learned in this practice and learn the output section. So in here, we give you a console, which is an R console by itself, and we include the codes that you have just learned in the previous sections. And we first give you the codes as such and explain it to you that you can simply run this code, and you are expected to understand, or you are expected to see some of the answers or some of the plots that is associated with this code that we have seen that it can produce in the learn the code section. So I simply run this code and see whether it runs or not. Yes, I see that the output tells the success in running the code, and the answer is correct. And I produce several plots, and I also produce an output of my principal component analysis variance. And I can open these files and see whether the output that I created agrees or goes very well, or described how it is being described in the learn the code section, because we are using the same input data. We can check that. Yes, okay, it does. Very good. So now you can start exploring different parts of this code and start fine-tuning and start changing some of the parts of this code so that you can expand your understanding of the code that you have learned so far. And some of the hints that we have given here, for example, if you see in this area where we are calculating the principal component analysis, we again have not used the option to scale or to center the data. So we can actually do that, and some of the example codes are given here. So you can try scaling and centering the data. You can now modify this by including scale dot equals true, and center equals true. And then if I run the code again, I can clear the console and the outputs by clearing it. And then if I let it clear, if I run the code again, okay, Now I have changed something in the data. So this checks for the pre-calculated data. So by centering and scaling, some of the outputs have actually changed, but that's okay. That's why my mm, mm, pop-up said success in running the code, which is what my aim is, but the answer is incorrect because this code block is intended to produce some other answer. So now I can actually check whether or not I see a difference if I perform scaling and centering in my original data. Yeah, in the principal component analysis or not. If you have saved this one, then you can easily check. And you can again come down and see how you can write the output, right? I just showed you from the server how you can access the principal component analysis that is calculated for your input file, and you can save it as a table. You can use the table for further downstream analysis like clustering and other methods. So this code block actually tells you how to save the table, right? And a necessary piece of code or a focused piece of code is actually given here, where you can find out how to write an output from our environment into a file. And again, we have to tell what to write or what object to write out and what is a file name and some associated characters, how this file name has to be or how this file has to be created by like, do we have to include column names? Do we have to include the row names, or how two columns have to be separated, just like how you have described the data while loading in the data. So let's run this and see for ourselves. Yes, in addition to a couple of plots, I can also see there's a table that is produced, and you can open this table and you can download this table, save it as download this table, and find out about this principal component analysis calculated for your input file. So let's time constraint, let's not do that. I will let you explore that. You can download this table, load up in Excel, and then explore this table further. And then after that, what we have actually done is now you got a good hang of how the lesson works and how you learned the code and how you can access or how you can use the R console to run these codes and even modify these codes to suit your requirements. And now in this section, we have deleted some of the syntaxes to keep you interesting. And then you have to find out where we have deleted the syntaxes and complete the code there to run this code successfully. So if you run it as such, it's going to throw you an error and error running the code. And most of the time, like in any programming language, the error message that R is going to give you is going to be very difficult to understand. So we clear this, and then we can go through this code line by line. If you had understood, or you can also refer to what we have done here and what we have given here, and find where the necessary completion is required for this. And I see, okay, I see that the principal component analysis function is not given in its full form. And I also see that there is a problem in plotting the principal component analysis. So let's complete it, PCA res. And I can also see that you have to run it time and again and see if this produces any error or if it's not producing any error. Now, I have rectified some of the portions, and there are some more portions that I need to find out where the error is. And one of the areas where the error is, it's here. And I have to mention which column corresponds to coloring or annotation in the data expression table. That is group. And if you can know that if you open it in another file or in another tab, you will know that. And let's see what else do we have. And we can actually see here, okay, in this plot, we have to mention, we have to enable label. And in this plot, we can mention, we have to disable framing. Okay, once you do that, we can again try running the code. Okay, this time I made all the necessary changes, and I get success in running the code, and the answer is correct. And I create a lot of plots, which is very what all the plots that we have actually explored. And that will help us understand the patterns in this data. So after this, after letting you complete the code, and we give you an assignment where again some of the parts are missing, and you can again explain or try running this. So I give you this as a challenge. So today you can try completing this one assignment, and let us know by email or by email if you can successfully finish this option or not. And for those who wants to know more or who wants to explore a little bit further than what we have learned so far, we have more options where we have another input table. My computer is frozen. Yes, we have another option for another input table, which will help you understand the power of principal component analysis in identifying outliers. So you can go beyond the basics here, where you will get a taste of what I have demonstrated you in the server. You take the output of the principal component analysis and then apply k-means clustering to that table to identify the different types of clusters or different number of clusters that your principal or your data contains, or your k-means clustering identifies in your data table.